Batrachotoxin, or BTX, is a cardiotoxic and neurotoxic steroidal alkaloid. It is one of the most potent alkaloids. This toxin was named and separated by John W. Daly and Bernard Wittkopf, who isolated the toxin from 4 other major toxic alkaloids. Batrachotoxin was first isolated from the poison dart frog. The toxin is released by the frog in response to agitation, pain, and when it feels threatened. It is secreted through the glands on the back and behind the ears of the frog. They do not have any method of injecting the poison in prey. The only way the poison produces the effects is when other animals eat the frog. These frogs lived in the Amazon rainforest. The poisonous species are often brightly colored to warn other animals of their poisonous nature. The poison dart frogs do not produce the toxin themselves. It is believed the frogs get the poison through the consumption of beetles and other insects in their native habitat. This has been proven through the captivation of the frogs. Frogs isolated from their habitat did not produce the toxin. This makes the toxin very difficult to study, as the poison dart frog is a threatened species, and because the frogs only secrete small amounts of the toxin. One study used only 10,000 poison dart frogs to produce only 180 milligrams of batrachotoxin. The golden poison dart frog is thought to be the most poisonous animal on Earth. The small 5-centimeter frogs have enough venom to kill 5 to 10 adult humans, or about 20,000 mice, as only 2 micrograms of the toxin is needed to kill a human or other large mammals. The golden poison dart frog has 27 times more batrachotoxin and is 20 times more toxic compared to other species. Other bird species in New Guinea also contain the toxin on their skin and feathers, such as the ifrit and the patui. The toxin is also believed to come from the beetles and insects that they ingest. The poison has been found in the Melbird beetles, which is a food source of these birds and the poison dart frog. An important use of the toxin is used in local indigenous cultures. The frogs are exposed to fire, which causes them to secrete the poisonous fluid. Arrows for hunting are then soaked in the fluid. The poison on the tip of the arrow is enough to cause instantaneous nerve paralysis in animals such as monkeys and birds. Other cultures also use a stick that is put through the mouth of the frog to the hind legs, causing the frog to perspire and emit a white froth. The arrows are then rolled in the froth and can be preserved for up to a year.
There are a few methods of absorption possible when it comes to the BTX toxin. These include oral, cutaneous, intravascular, and/or intramuscular methods. The most common mode of absorption is oral, where the predators of the frog or other BTX-carrying organisms ingest their prey and then succumb to the toxic effects as the toxin is passed through the digestive system and absorbed into the systemic circulation. The toxin may also be absorbed cutaneously, or through the skin, by any predators or otherwise who may have come in contact with the animal or any area where it may have been. The drug is largely lipophilic, as will be discussed later, which allows for its absorption into circulation through the outer skin. Lastly, the drug may also be absorbed through intravascular and/or intramuscular methods. This scenario is primarily relevant in South or Central America, where the indigenous people have been known to coat their weapons, mainly blow darts, with the toxin. When a dart punctures the skin of an animal, the toxin is thereby distributed within the animal by exposing muscle tissue to this toxin. The primary site of action of the BTX toxin is at the outer membranes of nerve and muscle cells, where the BTX molecules have a high affinity for the voltage-gated sodium channels. Once bound to these specific transport channel proteins, the channels are locked in an open state. This binding is irreversible, which adjusts a chemical and/or structural change to the sodium channel which locks it open. Since binding only occurs in open sodium channels, it has been hypothesized that the BTX toxin binds within the pore of the channel. The binding thus causes disruption of action potentials across the membrane. The cell is depolarized due to the influx of sodium ions after the toxin is bound. This also results in the potassium channels on the membrane to open and allow for potassium ions to continuously leave the cell. Usually the cells would then be repolarized in a continuous cycle of action potential. However, the binding of the BTX molecules halts this cycle and permanently depolarizes the cell. The consequences of this disruption will be discussed shortly.
Batrachotoxin, a steroidal alkaloid, has 2 main parts to its structure. The steroid part is lipophilic and closely resembles cholesterol, while the basic or alkaloid part is responsible for the toxic properties of the molecule. As of today, batrachotoxin's mechanism of distribution is still a mystery and is largely subject to research. This doesn't come as a surprise, since the majority of steroidal alkaloids, not only batrachotoxins, have yet to have their modes of distributions deciphered. However, There is speculation suggesting that the toxin follows many of the same modes of distribution as general steroids. The majority of the structure of steroids, like cholesterol, is hydrophobic, meaning they can theoretically be absorbed into circulation and distributed to lipophilic areas of the body. Experts mention that it is possible that the alkaloid part disguises itself in the larger steroidal part of the molecule, making batrachotoxin as a whole subject to many of the same transport mechanisms around the body as something like dexamethasone, a synthetic drug used primarily as an anti-inflammatory, with the main difference being batrachotoxin sports a basic nitrogen rather than acidic hydroxide. It can be inferred that the alkaloid part of the batrachotoxin helps the molecule as it is being transported through aqueous mediums, albeit to a minimal extent, as the majority of the molecule is still hydrophobic. The main importance of the hydrophilic part is to perform its toxic effect at the site of action. The molecule produces its effect on the outside of the cell membrane, which is conveniently polar for batrachotoxin. The hydrophilic portion of the molecule can orient itself towards the sodium channel to produce its effect. In the poison dart frog, batrachotoxin is found predominantly in dermal glands and is secreted to the skin when the organism feels its safety is in jeopardy. When the toxin is introduced into animals like humans, it almost selectively targets the sodium channels of the heart. This is important since if the sodium channels of the heart are irreversibly placed in their open conformation, the heart will no longer be able to depolarize and will be consequently rendered incapable of performing its most vital function of contracting and relaxing to pump blood throughout the body. The LD50 of batrachotoxin is a very low 0.002 to 0.007 milligrams per kilogram, which is a testament to the toxicity of the substance and its ability to reach its intended target and efficiently produce its effect.
Batrachotoxin's metabolism and elimination is difficult to study due to many factors. A main factor is the lethality of the drug. Not only does it cause death, but it causes death at such a low dose and with such a rapid onset that it is difficult to study in small animals and would be unethical to study in humans. Other factors of the limited research include the endangerment of the poison dart frog species, the inability of the frogs to produce the toxin in captivity, and the inability to synthesize the compound in the lab.
Batrachotoxin possesses many more toxic effects than it does therapeutic. When introduced to its victim, the toxin irreversibly binds and forces open voltage-gated sodium channels, causing immediate nerve and muscle depolarization. With these channels forced open, victims experience a combination of fibrillations, arrhythmias, paralysis, and/or heart failure. It is usually the respiratory paralysis that ultimately causes death. However, the most debilitating problem of all with this toxin is its irreversibility. Once bound, the sodium channels are open for good and cannot be reshut. As a result, once a victim has come in contact with the toxin, it is impossible to stop its wrath. The resulting paralysis and fibrillations are a cascading death sentence. Nonetheless, the toxic effects of batrachotoxin may not be all bad. Its highly specific mechanism of action on sodium channels proves to be of interest to scientists around the world in studying the fundamental processes of heart function and the sensation of pain. In fact, batrachotoxin has been studied extensively in labs to gain understanding of voltage-gated sodium channels and their structures, functions, and interactions with different proteins. In recent years, scientists have been able to synthesize batrachotoxin's chiral twin, which oddly enough has the exact opposite effect. Rather than forcing the sodium channels open, the enantiomer forces the channels shut, thereby preventing the outflow of sodium needed for nerve and muscle relaxation. Thus, instead of paralysis, this new molecule causes contraction. But also, unlike the original batrachotoxin, it is reversible. This discovery opens the door to many new possibilities of pharmaceutics to combat pain and nerve disorders. A couple of examples include congenital insensitivity to pain, or CIPA, which involves the inability to feel pain or temperature, and erythromelalgia, or the man on fire syndrome, which involves the feeling of burning alive from the inside. Both conditions have no known cure at this point, but the synthesis of batrachotoxin and its enantiomer could help scientists gain a deeper understanding of their mechanisms of action and hopefully one day come up with a drug that can counteract their devastating effects.
In order to facilitate this research, new policies must be implemented to protect the endangered poison dart frog species. Although scientists are close to being able to synthesize batrachotoxin on their own, they are not quite at that point and need the frogs to continue their research. Therefore, unless the poison dart frogs keep populating or artificial synthesis of the compound is discovered, this cutting-edge research will be at an indefinite halt.